This video is going to take you through the second half of chapter 24. This half is going to talk about metabolism. All of these terms we defined in AMP1, so just pause it if you need the definitions. So, next you have describe cellular respiration and provide the chemical equation for glucose metabolism. So cellular respiration has different names. First thing you need to remember is it is entirely different from what you typically think of as respiration, because most people tend to tie that word to breathing. You breathe so that you can do cellular respiration. Glucose metabolism is going to be the way that we're going to start with this, but you can input other molecules into metabolism. It's just easier to start with glucose. The whole reason that cells do glucose metabolism or cellular respiration in general is so that they can make ATP. I will mention that we dipped our toe into the topic of cellular respiration last semester in Chapter 9 when we were talking about how muscles regenerate ATP. There were 3 methods: creatine phosphate, glycolysis, and then cellular respiration. So we had briefly talked about this. Now we're going to talk about it in a lot more detail though. For provide the chemical equation for glucose metabolism, whichever one of these 2 ways explains it to you more, this is it. You have a digestive system to supply you with glucose. You can feed in proteins and lipids to this, but we have to modify those first before we can put them in here. We're essentially going to be burning the glucose or the major nutrient, and then oxygen is going to be the terminal electron acceptor that receives electrons from electron carriers that strip electrons from whatever our precursor molecule is. If we don't have oxygen, we can't do this burning process that's going to be happening. We're going to end up doing this to produce the ATP, but we do also produce some carbon dioxide and some water in the process. So respiration, as talked about in the respiratory system, you ventilate so that you can get oxygen into your bloodstream so that you can eventually take it to your tissues. This is why you need that oxygen. When you die because you're not breathing, you die because you can't do this and your cells run out of ATP, and the cells die if they don't have enough ATP. Now, the number of ATP that you see in this top one, the number is different in every textbook that has ever been published. They used to say it was 38 ATP. That is a theoretical maximum. Most cells are closer to 20 to 24 ATPs per glucose molecule, so don't focus too much on what this number is right here, because it varies depending on what else the cell is doing at a given time. Next you have this figure. You're filling in any of the terms that I have. You already have some terms in the picture in your notes. This is just helping you fill it in. So depending on who you ask, there's either 3 or 4 steps. There's glycolysis, which is going to start with glucose and end with 2 pyruvic acid or pyruvate molecules. The pyruvate runs through the transition step where we generate a couple of acetyl coenzyme A's, or acetyl-CoAs for short. Those acetyl-CoAs run into the Krebs cycle, which is also called the citric acid cycle, which is, so I think your textbook is merging these 2 things into one step, the transition step and the Krebs cycle. I tend to like to keep the transition step separate, but I don't think your book does that. So this is one step according to your textbook. The whole point of doing these first 2 or 3 steps, depending on who you want to believe here, is so that we can generate these electron carriers, either NADH or FADH2. These molecules are going to bring electrons to what I still call the electron transport chain. It's also called oxidative phosphorylation or chemiosmosis. It has a bunch of different names, but I'm going to call it electron transport. This is where we generate most of the ATPs that are going to be generated from a glucose or a protein or whatever. But again, don't focus too much on the numbers. This is still getting you a high number. The only numbers that hold true are, we're going to make a total of 4 ATPs during glycolysis, but we have to pay ourselves back 2, so we get a net gain of 2 ATPs. Each spin of the Krebs cycle gets you 1 ATP, but we spin twice for each, once for each molecule of pyruvic acid, or twice for each glucose molecule. So we get 2 ATPs per glucose. And then the electron carriers bring in their electrons. This is where the variability comes in. It can be as low as like 16 ATPs. It can be as high as 32 ATPs, depending on what textbook you're looking at. But this is where it's not a one-to-one relationship. It varies depending on what else is going on in a cell at any given time. All right. This video is from a non-majors textbook. It's kind of going to give you just the overview of what's happening here, so hopefully this will help you out.
As this mountain biker heads up the trail, the breakfast he ate this morning is being burned to power his bike ride. His breathing rate increases as his leg muscles demand more oxygen to burn more fuel. Let's zoom down to where this fuel is burned, our cells. Here, the blood vessel on the left delivers fuel and oxygen to a single muscle cell. In cellular respiration, energy in fuel is converted to ATP, shown here as starbursts. Most ATP is made in the cell's mitochondria. ATP powers the work of the cell, such as contraction. Let's take a closer look at how ATP is produced from a molecule of glucose, our fuel. Only the carbon skeleton is shown to keep things simple. The first step is called glycolysis, and it takes place outside the mitochondria. To begin the process, some energy has to be invested. Next, the molecule is split in half. Now, the molecule NAD+, an electron carrier, picks up electrons and hydrogen atoms from the carbon molecule, becoming NADH. Keep track of the electron carriers. They play an important role by transporting electrons to reactions in the mitochondria. In the final steps of glycolysis, some ATP is produced, but not much. For every glucose molecule, only 2 net ATPs are produced outside the mitochondrion. However, glycolysis has produced pyruvic acid, which still has a lot of energy available. Let's follow this pyruvic acid molecule into a mitochondrion to see where most of the energy is extracted. As the molecule enters the mitochondrion, 1 carbon is removed, forming carbon dioxide as a byproduct. Electrons are stripped, forming NADH. Coenzyme A attaches to the 2-carbon fragment, forming acetyl-CoA. Coenzyme A is removed, and the remaining 2-carbon skeleton is attached to an existing 4-carbon molecule that serves as the starting point for the citric acid cycle. The new 6-carbon chain is partially broken down, releasing carbon dioxide. Several electrons are captured by electron carriers, and more carbon dioxide is released. The carbon dioxide that you exhale comes from the reactions of cellular respiration. 2 ATPs are produced by the citric acid cycle for each molecule of glucose. At this point, only a small number of ATPs have been produced. However, more energy is available in the electrons that are being transported by electron carriers. While the citric acid cycle starts another round, let's follow an electron carrier to the next step in the process. Electron carriers such as NADH deliver their electrons to an electron transport chain embedded in the inner membrane of the mitochondrion. The chain consists of a series of electron carriers, most of which are proteins that exist in large complexes. Electrons are transferred from one electron carrier to the next in the electron transport chain. Let's take a closer look at the path electrons take through the chain. As electrons move along each step of the chain, they give up a bit of energy. The oxygen you breathe pulls electrons from the transport chain, and water is formed as a byproduct. The energy released by electrons is used to pump hydrogen ions, the blue balls, across the inner membrane of the mitochondrion, creating an area of high hydrogen ion concentration. Hydrogen ions flow back across the membrane through a turbine. Much like water through a dam, the flow of hydrogen ions spins the turbine, which activates the production of ATP. These spinning turbines in your cells produce most of the ATP that is generated from the food you eat. The process you've just observed, cellular respiration, generates 10 million ATPs per second in just one cell. That ATP can power a biker up the trail, or it can power your brain cells as you learn challenging biology topics.
Alright, so hopefully that gave you an idea in your head about how glucose metabolism is going to work out. Because your cells are primed to use glucose as their primary fuel source, it's important that your body maintains blood sugar levels. And so the next few words that you have, students have a habit of calling them the big G words, but don't do that. Make sure you try to say the name. Make sure you pay attention to the Latin roots. So if you break this word down, genesis is to make. Glycogen is what we're actually going to be making here. And so anytime your blood sugar gets to be too high, remember you release insulin. Insulin promotes the liver to take all the extra glucose and then store it as glycogen inside the cells. And so this is a process that lowers your blood sugar level, but at the same time stores that extra sugar, but not as sugar. It's sort of storing it as your version of starch. So glycogen is the animal equivalent of starch. Now this is the exact opposite. If you break this word down, lysis means to break or to pop, and then glycogen is glycogen, that starch that we were just talking about a second ago. So glycogenolysis breaks down glycogen, releasing glucose molecules. The hormone glucagon promotes this activity, especially in the liver, and so this is what would happen anytime your blood sugar level was starting to get too low. You would start to break down your glycogen stores to keep your blood sugar up where it needs to be so that you can carry out cellular respiration. Now you got another last-ditch effort to try to maintain your sugar levels. Gluconeogenesis. If you break this word down, neo is new, genesis is to make, and gluco means glucose in this case. So we want to make new sugars. This is essentially, you can convert proteins and fats, which are your other major nutrients, into sugars in your body so that you can maintain your blood sugar level where you need to. This is another thing that glucagon can promote when your blood sugar level gets to be too low. So it's another way for you to elevate your blood sugar level. Now, you can use some other molecules to feed them into glucose metabolism. It's just that we're not going to be starting with glucose. So some of the stuff you need to know. What do the lipids get broken down into? Glycerol and then fatty acids. The glycerol is a 3-carbon chain. We can turn that almost immediately into glyceraldehyde 3-phosphate, or G3P is what it's usually known as. And then that can get fed into glycolysis, where it'll get converted into pyruvic acid, and then we can continue on through the transition step, the Krebs cycle, and then electron transport. So the glycerol feeds in pretty easily. The fatty acids, on the other hand, they're these big long chains of carbon and hydrogen. What will happen is, instead of trying to feed in like a giant 28 carbon molecule, we'll split it so that we keep stripping 2 carbons off the fatty acid chains. Well, a 2-carbon chain is an acetyl group. So we can feed that in at acetyl coenzyme A down here in the transition step, because when we break that down as a 2-carbon group, it becomes an acetyl. All we have to do is add coenzyme A to that and it becomes acetyl coenzyme A. And so the fatty acids get fed in at the transition step. So the lipids feed in different places depending on whether it's the glycerol or the fatty acids. Now, one of the other things that I like to mention at this point, because there's such a vast amount of carbon that's associated with fats, fats are worth more than twice the calories per gram than glucose, or proteins for that matter. So lipids are worth 9 calories per gram. Both glucose and, carbohydrates in general, and proteins are only worth 4 calories per gram. And so part of the reason why that is because we can feed them in multiple places, and we can just get so many acetyl groups out of a fatty acid. It's just worth more. Some problems that are going to go along with this. Anytime your body is not using glucose, there are all these chemical reactions that you have to do to modify things so that you can feed them into cellular respiration. Now, when you're using lipids as an energy source, your body is going to start to make ketones. When your body is making ketones, you are in ketogenesis. Now, there is this fad diet that is going on right now that's called the keto diet. The keto diet does promote ketogenesis. If you do the diet correctly, it can be beneficial. You can lose weight. You might not feel as hungry while you're on it. But there is a problem with this. If you don't maintain your ketone levels very well, they're acids, and so it drops your body's pH, and then you're going to be excreting those ketones in the urine. So whenever you are excreting your ketones in the urine, you are in a state that is called ketosis. Mild ketosis, not a problem necessarily, but again, if you're not doing this right, your blood pH starts to drop, your brain really hates that, you can go into a coma and you can die. And so ketosis is not something you should be playing around with if you don't know how to properly maintain the levels. And the problem with this is most people who are on these diets, they're following bad examples and they have no idea what their ketone level is, and so they don't know if they're in a dangerous place or they don't know if they're in a fairly mild state of ketosis where they are actually losing weight. Alright, now metabolic acidosis ties back to what I was just saying. If you're accumulating ketones in your bloodstream, you are dropping your body's pH, and you are doing that as a result of metabolic reactions, and so that's metabolic acidosis. This is in contrast to respiratory acidosis, where you were building up carbon dioxide levels, so you were dropping your pH. So that was a breathing problem that dropped your pH. Now it's a metabolic problem that's dropping your pH. Again, end result still the same, brain's unhappy, coma, death, so we don't want to play around with acidosis at all. Keep your body's pH where it's supposed to be. The other little side note that I have here is if a person is in metabolic acidosis, either because they're on a stupid diet, they're not managing their diabetes very well, or they are literally starving to death, they will very often exhibit fruity smelling breath. I've heard it described as like the person is chewing juicy fruit gum even though they're not. So if you're ever dealing with a person who seems confused or unresponsive, or they're not doing what you're telling them to do, and you notice they smell kind of like juicy fruit, they're probably in acidosis and they can't control their behaviors very well because their brain is dying on them. And so they need medical attention now if you ever notice that. All right, for proteins, how do they get fed in? So remember, proteins are made out of amino acids. Amino acids have several different groups to them. They are carbon and oxygen and hydrogen, but they do also have that amino group, which is an NH2. The nitrogen has no place in cellular respiration at all, and so that nitrogen group has to be removed. When it's removed, it gets converted immediately to ammonia, which is NH3 that you can see right here. Ammonia is fairly toxic. Your body does not like that. And so your liver converts the ammonia to urea, which is a little bit less toxic. And then your kidneys are going to get rid of the urea. That's why they make urine. It's because of the urea that's in there. So again, there's some additional steps that we have to do if we're going to use proteins to feed in here. Where the amino acids get fed in, it depends on which amino acids you're looking at, but generally speaking, they're going to get fed in during glycolysis or during the Krebs cycle. They don't really go into the transition step very well. This is just the explanation of deamination that I related to you guys earlier. So remember that the amino group has to be removed from the amino acid. That's what deamination is. Immediately after it's removed, it becomes ammonia, and then the ammonia gets converted to urea in the liver. All right, after that, compare and contrast the absorptive state with the post-absorptive state. You're essentially just going to be getting down what this says right here. So the absorptive state is also called the fed state. If you have eaten, recently, you are in the absorptive state. Because you have the nutrients that you need during this point, most of your cells are going to be using glucose metabolism to keep their energy stores up. You're going to have plenty of vitamins. You're going to be storing excess nutrients in the body. Basically, the body is happy during the absorptive state, and it's storing things because it knows that you won't be in that state for very long. The post-absorptive state is also called the fasting state. If you haven't eaten in 3 hours or more, you are probably in the post-absorptive state. At this point, you start breaking down the stored nutrients that you had created earlier. So your extra glycogen, you start to break it down. You start to take some of the lipids and proteins and you do gluconeogenesis with that so that you can generate more glucose. At this point, your body switches more to we just need to maintain our blood sugar levels, because your brain cannot use anything but glucose as an energy source. And so we have to maintain that blood sugar or your brain starts to die. Some of your other cells, though, they can switch to other pathways and still survive just fine. It's just that we've got to maintain that blood sugar to keep the brain nice and happy. How does the liver help with all of the followings? Table 24.7 gets you all of the stuff that you're going to need for that. We've pretty much mentioned all of these things before, but just remember you can always pause it to get what you need. We've said all of that. We've said this stuff here except for the lipoproteins, which we're coming to here in just a second, but we've said most of the rest of that in other places. Protein metabolism, we've already said the whole deamination and all of that jazz, so we were all good with that too. Transamination I hadn't talked about and won't in the future, so I will slow down for that one. Transamination means that your liver has the ability to convert some amino acids to different forms when those other forms start to get too low. You can't, again, make all amino acids, but you can make quite a few of them. And so transamination converts one amino acid to a different amino acid, and your liver can do that for some. We store some vitamins, we store some minerals, and then we can transfer a whole bunch of things. We pretty much talked about all of those things before, so I'm just going to skip right past that. Okay. One of the next issues comes from things that your body consumes and your body needs, but you have to deal with some things. So lipoproteins are proteins that have fats attached. Lipoproteins are going to be shuttle molecules, especially for the movement of cholesterol. What happens is When the lipoproteins get to your liver removes the cholesterol and then sends the lipoproteins back out to the digestive system so it can pick up more cholesterol and take it back. That means your lipoproteins can be found in two states. The state where there's cholesterol attached is LDL. The state where it's unloaded the cholesterol is HDL. If you have more LDL, it means you're eating so much cholesterol that it's not able to unload everything into the liver like it's supposed to because there was already stuff there. So if you have high LDL, your book tells you to think of it as the L stands for lousy. It doesn't. It actually stands for low density lipoprotein. But LDLs are the bad cholesterol because it means you're not doing cholesterol homeostasis very well. You are consuming too much cholesterol. If you have higher HDLs, on the other hand, that means you are getting the cholesterol out of your body as fast as you're taking it in, essentially. So you're recycling things like you're supposed to. So you want to have a high HDL level but a low LDL level. If it's reversed, it means you could be building up those atherosclerotic plaques that we had talked about before. And so that's not good for your circulatory system. Remember, that increases the risk of stroke, heart attack, hypertension, a bunch of other problems. And so good, bad. What is the recommended total cholesterol level for adults? You're supposed to keep yours to less than 200 milligrams per deciliter of blood. If it gets to be over 200, even at 201, doctors at this point like to throw drugs at you. But the best way you can lower your cholesterol, eat right and exercise. It does way more than any drug, and it has only good side effects. And so please, diet and exercise. Statins are the drugs that doctors like to throw at you so you can lower your cholesterol levels. Lipitor is one of the highest selling brand name drugs that's out there, but that's what it does, is it lowers your cholesterol levels. What is meant by energy balance is the next thing that you guys have. Just like any other balance, it means energy in equals energy out. If you are in energy balance, it means you are not gaining weight, you are not losing weight, you are maintaining what you have. If you're in positive energy balance, it means you're consuming more calories than you're expending. You're going to be putting weight on, gaining weight, potentially becoming obese if you keep that up for an extended period of time. If you're in negative energy balance, you're going to be losing weight. Now, I am a math person, or at least I used to be way more of a math person, but this to me speaks better than a lot of other things. If you are having trouble maintaining your weight, you need to do a food log, and you need to see how many calories you're actually consuming during the course of a day. You can look up what your RDA is supposed to be, or there's this, RDA is for all nutrients, the EER is the estimated energy requirement, and it shows you how many calories a person is supposed to be eating during the course of a day. If you're over 18, you have to do a little bit of math to figure it out. That's what the little superscript there says, but if you take a nutrition class, they'll tell you how to calculate that. There's various apps that can also try to figure that stuff out for you guys at this point. But what I want to point out is basically nowhere on here does it say 2,000 calories a day. Here's where there needs to be a huge disclaimer though. Everybody is different. 2,000 calories for one person might make them lose weight, but it would make another person gain weight because everybody's different. People who are shorter don't need as many calories. People who are skinnier don't need as many calories. People who aren't breastfeeding or pregnant, they don't need as many calories. If you have a crazy high exercise routine, you need more calories than a person who just sits around teaching classes all day in her home. And so these numbers are recommended for people who are average height, average weight, average health, no sort of health problems, no disease states. And so that doesn't really fit for a lot of people. And so yours should be a little bit more personalized than what this is showing you. So energy in is everything that essentially you put into your mouth during the course of a day. It's all the food you eat, snacks included. It's all the drinks you drink, alcohol included. All of those things are putting calories into your body. Energy out. Some of that just happens because just breathing, you use some energy. Digesting some food, you use some energy. But you put more energy out if you lead an active lifestyle. So essentially, the more you exercise, the more energy you're allowed to intake. And so if you really like cheesecake, as I really love cheesecake, it means you better get off your butt and go do some exercise so you can eat said cheesecake. Now what they're doing in him is they're measuring his energy output. That will be able to tell them how many calories he should intake if he wants to maintain where he's currently at. So this is something that can be measured. It's not super fun, but it can be done. This one I wanted to pause on for just a second for you guys. Different books give you different formulas for calculating your BMI, which is body mass index. I think your book gives it to you using the metric system, but here it is in pounds and then in inches, your height in inches. If you're 5 foot tall, you're 60 inches. So if you're 5'6", you're 66 inches, but figure out where you are. Go ahead and calculate your own. National Institute of Health does have a BMI calculator. I've told you, so that's how it's calculated. Body mass index tells you, it's essentially what is your weight in relation to your height, and are those two things within parameters for reduced disease risk. It is not a perfect measurement system at all. People who are like bodybuilders, it will tell them they're obese even though their body fat is like 1% or whatever. And so it's important that you note that it's not differentiating between muscle mass and then fat. It's just height versus weight. And so it can give you some bad information sometimes. But for the average person, this shows you, are you at risk of different diseases based on your current height and weight. So 18 to 25 is considered to be normal. Normal means you're not at an increased risk of metabolic disease, of diabetes, of stroke, of heart attack, any of those types of things. If you're underweight, you're at increased risk of having malnourishment issues because you're not getting all the nutrients that you need during the course of the day. You might have weaker bones because you probably can't do a lot of weight-bearing exercise to strengthen them, so your risk of osteoporosis gets up. If you're overweight, you're at a slightly elevated risk of diabetes and heart disease. If you're obese, you're at a significantly higher risk of diabetes and heart disease. And so the different categories tell you different things. I will also say there are now additional categories because Americans have gotten so fat. We now have super obese and morbid obese, and so those are actually above this one. So yay, congratulations. We needed new categories because we're just that fat. Basal metabolic rate. This is, like it says in that definition, this is just what it costs in terms of your calories just to stay alive. This is not how much do you need when you're exercising. It's if you're just sitting in a room that has a good ambient temperature where you're not hot and you're not cold, you have not recently eaten so you're not digesting food, you're not having a panic attack so you're not breathing faster, you're just sitting there comfortable resting, that would be your basal metabolic rate. It does get a little bit lower in sleep, but this is just essentially the bare minimum that you would need to not die. After that, your book starts talking about other stuff. We get into temperature regulation for just a second. Normal range of temperatures, it depends on where you take the temperature and what kind of a device you're taking that temperature with. So generally speaking, there's two different ways you can take a temperature. You can take it from the outside of the body, that gets you, pardon me, the shell temp off down here, or you can have a probe inside the body somehow and that gets you the core temp. Core temperature is higher than shell temperature. So if you're doing this type of measurement, like a lot of doctors' offices have those temporal scanners now where they just run it over your forehead, that would not give you a 98.6 for the average person because your temperature on the skin is not supposed to be 98.6. Your core temperature, on the other hand, a rectal temp, if you do stick a thermometer up there, you're getting closer to the core of the body, so the temperature would be higher. So if you got a 99 for a rectal temp, that's perfectly normal, not a fever, because that temperature should be higher in that place. This down here is just showing you different temperature ranges that are considered to be average. One of the things I told you guys last semester was if you have never measured your temperature when you're not sick, you need to do that. Like when I am not sick, my temperature is usually in the 96s. So a 98 is actually high for me. Everybody is different, kind of like with everything else, and so you need to know where your normal is. Next up, different ways that you can exchange heat with the outside world. These are just definitions for you guys to get down, but you do just lose heat through radiation. If you've ever been next to a person, you can feel the heat radiating off of them, and so that's that. Conduction is when you touch something, like in the summertime, after my dog has been outside for a while, he likes to go to the bathroom because the bathroom has ceramic tile versus the rest of my house has hardwood floors. You can actually exchange more heat with a ceramic tile than you can with a hardwood floor. So that's him trying to cool himself off through conduction, not that he would know that. Convection is a little bit more complicated. Y'all have all heard at some point that warm air rises. So you're constantly producing warm air. As it rises, it gets replaced by cooler air. And so that's a way for you to maintain temperature. And then there's the whole sweating thing. When you lose water, you also lose the heat with that water. So that's evaporative cooling. Hypothalamus does help you regulate your temperature. Okay, so if your body temperature drops, what are you going to do? The other way to ask this is, you're cold, what are you going to do about it? Most of these make sense. You know if you're cold, you're going to shiver. You know if you're cold, you tend to turn paler. You turn pale because you constrict those cutaneous blood vessels. What you're trying to do there is keep your blood closer to the core where it's warmer, because if it goes closer to the outside, you're going to radiate off more of your heat, and that's going to increase your cooling, and you don't want to do that. And the other two things really go together. You're going to increase your thyroid gland's activity levels, so you're going to release more thyroid hormone, and then that's going to increase your metabolic rate so that you're generating more heat in your body. Okay, exact opposite question. Now you're too hot. What are you going to do about it? You know you're going to sweat. You know you tend to get red. You get red because you dilate your cutaneous blood vessel. Now, because you're hot, we want to radiate off some of that heat, so we're going to take the blood closer to the surface so you can do that. The last thing your book mentions is behavioral changes, and I'll go back to my dog who likes to lay on the cold floor. He doesn't do that in the wintertime. That's a behavioral change. When you're too hot, go inside where it's air conditioning, eat some ice cream. You know, you can do lots of different things behaviorally to try to keep your temperature where it's supposed to be. Hyperthermia means that you are too hot. If you stay that way for too long, you will sweat so much that your blood pressure actually starts to drop, and that's where heat stroke comes in. Heat stroke in Texas kills people every single year, and it's because they don't maintain their water and electrolyte balance like they're supposed to. So if you're ever working outside in the summer heat, please make sure you're cooling off periodically and drinking plenty of fluids so that you don't get heat stroke. Hypothermia is the exact opposite. A lot of people think this isn't an issue in Texas, and yet it very much is. We have a homeless population, and it got down into the 40s last night before I recorded this. That's enough to put a person into hypothermia. You can end up losing fingers and toes as you cut off blood supply to those regions to try to keep your temperature okay. So this is still a problem in our area. Last up for metabolism, there's an interesting disease. It's not super common, 1 out of about every 10,000 births. It is today called PKU, Phenylketonuria. So this has to do with one of the specific amino acids. It's called phenylalanine. Remember, amino acids make proteins. The first ever newborn screening test was to test for phenylketonuria, because if you feed a person with phenylketonuria a regular diet, what happens is as they break down their proteins that have phenylalanine in them, they can't break down the phenylalanine, and so it builds up to toxic levels. It ends up destroying the brain, and they become more and more and more mentally retarded as time continues to go through. So by knowing as a newborn that the child has phenylketonuria, we can put them on a special diet, which is a crap diet. Nobody in the world wants to be on that diet. But if we put them on this diet, they don't develop that mental deterioration, and they can be fully functional adults. In fact, there is now a medical doctor who has PKU. She's had to stick on that diet her whole entire life. And so this used to be a condition where your kid would have it, and then you'd end up having to give your kid to some horrible, horrible home for challenged children, or what they used to call the feeble-minded back in the day, where if we'd have just known about it and we could have put them on this diet, they would have had perfectly normal lives, but we just didn't know. Now, the other thing that you guys may have seen is if you've ever been looking at like sugarless gum or a Diet Coke can or something, one of the artificial sweeteners has phenylalanine in it. And so those foods are required by law to have a phenylketonuria warning on them. And so if you ever have one of those things, flip it over and then look. Now, I think it's aspartame. I always forget which one it is. Do I have a picture? Nope. Don't have a picture. But it has that on there, and so it has to warn anybody who has PKU so they know not to consume that particular food item. Because you would think sugar-free gum wouldn't have a lot of protein, but that sweetener has that phenylalanine in it, and so it can cause problems for them. Let's see. Said that. Anything else that I wanted to mention there? Not really. Okay, so usually the assignment that I like to give for metabolism and stuff is for everybody to do a food log, but nowadays there's not really a good app that tracks all the nutrients that is free for you guys to use. So I'm not sure what the assignments is going to be yet for this chapter. Just be on the lookout in assignments.